Hi everyone, welcome to today's live interactive session for final week for this course, week 12. Uh, Dr. Robin is here as well.
Hi.
Hi. Uh, there are a couple of questions in the Google Sheet. Should we address those?
Uh, sure,
Uh, would you like me to paste them here in
if you don't
the?
mind. Yeah.
Yeah. Sure. In case you all have any other questions, you can type them in the chat box as well.
Yeah, that's an interesting question. No, I think there's a question about the human genome and the function of each gene. Unfortunately, that's not how it works. There's a lot of interactions that happen. So yeah, for some, this is the golden uh, that's the idea. That's what one would like to get to, and probably one can. But yeah, but otherwise, uh, uh, I think we are quite far from that. Yeah, frequency spectrum. I can explain that. Just give me a little bit. I'm trying to. I'll find the slide and then I'll explain that. Uh, uh, from Anurag Sharma. So in the Sanger sequencing, don't we have to correct for the base? Actually, that's because there is a die. So the one that gets added is specific. So that happens, but the base that gets called is fairly accurate. Actually, Sanger, there are errors, but although it's such a dated technology, it still has very low errors relative to, let's say, the next-gen sequencing or something like that. From Gajnan, there's a question about structure and how they are drawn and interpreted. The structure plots are basically, it's alleles that are shared between populations, I mean, between individuals in a population. So in effect, what it means is that there is gene flow if these alleles are shared. Uh, um, so, so that's how this is very popular in understanding, especially the tiger gene flow. I don't think any other species has been so as extensively studied as tigers. We've done a little bit with birds, but otherwise, large scale data on animals of the same species is not that common from India. So yeah, so how is it drawn? I mean, so there are analytical packages, and what you're seeing is a representation of that. You know, those bars that you saw. So if you have a bar that is just only in one location, one color bar, let's say orange, all the orange in Bangalore is, all samples you get from Bangalore have just some specific alleles. But if Bangalore and Chennai share alleles, that means that there is some gene flow. So let's say, you have orange here and then you have green there. So if there's a mix of green and orange in between Chennai and Bangalore, that means that these two are probably mixed. So that's the graphical representation. So I hope, yeah, I mean, these are very, very advanced topics. even side frequency spectrum, I'm not sure. It's really advanced. All of these are advanced topics. So I wouldn't worry too much if you don't understand too much of that. But just understand that the whole idea of this that lecture was just to kind of indicate that all of these are possible. Uh, yeah, I think Akshay says that some of the tree diagrams, some the things were not explained. Again, uh, I'll have to find that figure, and then I'll project that and I'll show you. I'll explain that. Exam center, I don't know about that. What is haplotype diversity? Uh, Akshay asked what is haplotype diversity. Yeah. So if you think of genotypes, like genetic diversity, let's say, and if okay, so Sanger sequencing. So Sanger sequencing has a string of ATGC, let's say 100 or 200 base pairs. And then if that, you can call that one haplotype. And then if that haplotype occurs again, so there is no diversity, it's just a repetition of that haplotype. But if you have a few bases different, so then that's a different haplotype. So that is basically haplotype diversity is at the heart of genetic diversity. Uh, yeah, I realized that what I'm saying is not going to make too much sense. Just a moment. Yeah, sorry. Yeah. Uh, where was I? Yeah. So I realized as I'm seeing a lot of the questions that I think that I'll explain it, but I don't think many of you will actually understand it too well because these are topics that I think need longer engagement. I mean, you actually need a full uh, conservation genetics or what I summarized was from probably an entire semester or something like that into one lecture. So it's a bit unfair on all of you, but the idea is to just introduce you that there are all these things that are possible. Uh, Hopefully, we'll have another real course where you'll probably learn some of those ideas. Uh, yeah, if the island, I'm just saying the next question from Abhishek Guha. If the island is small and closer to mainland, will it have lower extinction and greater immigration? Yes, that it would. We did that in the previous biogeography module as well. Doesn't have much to do with genetic diversity as per se, but of course, genetic diversity will also increase if populations are connected. And small populations means that small isolated populations have, uh, what's that called? Private alleles and things like that. So, yeah, uh, it's just a moment. Let's put on. Yeah. In Sibley's, how the relationship with hybridization temperature. Yeah, Akshay asked about this Sibley's DNA hybridization technique. Uh, you know, if you understand how a PCR reaction works, where you're trying to get one strand of a primer to attach to some other DNA. It's not very different from that. It's just that what they were doing was directly they were trying to see how much of how much hybridization happens between different the it's not hybridized as in individuals hybridizing. It's just the genetic hybridization. Yeah. So and that happens at different temperatures and to different extents. And what they said was that the more similar they are, that there is this hybridization thing happens more. Let me again try to find some a reference for this that you can look. Just give me a moment. I have something that I just share screen. Yeah. So you have the DNA. This is from some random thing that I just found. So pardon all the background ads and everything. But this is what happens. Basically, you have the two different strands of DNA. That is what is hybridizing. And when so organism A and organism B, you heat them kind of together, which is what happens in all your PCR reactions as well. And the extent of hybridization, this is what it is. So complete hybridization is when organisms are identical. Partial hybridization, that means that they are somewhat related, and no hybridization that they are unrelated. Yeah. So I mean, it's a very crude thing, when they were still working out. This was without sequences, without knowing what is the ATGC and so on. They don't know the basis. They're just putting the things together, heating it up, and then kind of seeing if they're hybridized or not. Of course, they had the idea that there are those base pairs that are there, and this is the chemistry. So they are working with that assumption, but without knowing the exact base pairs that are there. So today it's considered very rudimentary, but I hope you understand that, given the history of how we did bird taxonomy, particularly, uh, yeah, that was important. Drumming sound or acoustic feather sound is considered a song. Yeah, it's very difficult to find these definitions of what is song, and especially for non-passerines. For a lot of the passerines, it's straightforward, but for the non-passerines, it's just uh, uh, yeah, it's just crazy. I mean, a lot of things are called songs. Yeah. Sorry about that, Sushila, but that's just how it is. Yeah. Gaurav, in experiments such as a forest solid phylogenetic experiment, how do we assign the evolutionary timeline to the molecular data? Are museum samples, stuffed birds, uh, used in cases where historical sequence data is not available for comparison? Yes, the short answer to that is that we discussed in the last last discussion session as well. Uh, there are different ways to do this, assigning the timelines. The simple one is that you assume that there is a clock-like mutation that is happening. That means that the mutation accumulation is happening very periodically. So people have said, yeah, uh, uh, 2% per million years or something like that. So if you kind of say that there is 2% change that happens every million years, then you can look at what percentage of difference is there between your two target taxa. Suppose it's 10%. So then you can kind of draw back saying that, okay, so that means that it's 5 million years of divergence. And this is literally what we started out with when we were doing the Shulikola phylogenetics. That was the first thing that we did. And then, of course, there are ways to improve that. So that is with a phylogenetic tree, when you can say that I know that there was, let's say, an ancestor of all owls at some point in time, which is being dated independently using, let's say, carbon dating or something like that. And that you can kind of, in your phylogenetic tree, you can constrain your relationships. You can say that uh, in the tree, this is the location at this point of time. There was this fossil, and we know that it is ancestor to all owls because of various features that were there or something like that. So then, given that constraint that you have put, what is the relationship? I mean, sorry, not relationship, the timeline. So you can kind of you can use very complicated models and use fossils and things like that to get your time estimates. Uh, are museum samples used? Yes. So even in the Shulikola tree, we used a few museum samples. That's not very difficult to do, and technology is getting more and more advanced in doing that. Sorry, I'm just trying to find the the mm, yeah the slide for the other one, but yeah. Anyway, in cases where the sequence data is contradictory to existing taxonomical placement, which was possibly based on physical attributes, how are such conflicts resolved? Yeah, so once again, I think that the Sholikula is a very good example of this because when I was working on it, it was thought to be a short wing, and short wings are in the Himalayas and supposed to be related. But when you look at the phylogenetic relationships, it's actually not related to the short wings. It's some other flycatcher. So uh, so yeah, uh, the 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 genetic data, there is usually not so much of a problem with genetic data and trusting genetic data when it comes to deeper phylogeny, because there is no other way to resolve that. That is the best that is possible. But if you think about differences between subspecies, if you have two different subspecies or three different subspecies, um, and people think that it is one species or something like that, And that, if you try to split with only phylogeny, that is where you run into problems, where physical attributes say something, but your genetic data says something else. This is more difficult. And in this case, what I described in the other session as well was that we need to use multiple lines of evidence, which is use genetics, use acoustics, and morphology. So uh, essentially, it's trying to say that let's use the best of the data that is there today, and then you come up with the relationship that you see. Uh, and I have to say that saying genetics is seems like a simple one word, but there is all kinds of genetics. As you'll see from the questions itself, that we started out with DNA hybridization, and we went on to Sanger sequencing at some point, and then there were these barcoding genes that were proposed for a while. That was a big rage. Slowly, we started going from one gene to multiple genes, and now, usually you need thousands of genes. And slowly, people may say that you need a large part of the genome. So, how much data is actually enough to call something a different species? I mean, that's maybe something philosophical as much as technical. But as we understand organisms better, I think we should be able to use more lines of evidence and data to understand the diversity that we have around us. I hope that's kind of clear. Uh, I see the question from Pratik. He says that from the drumming sound, I have a question. In your lecture, you mentioned from the drumming sound of woodpeckers, we can identify the species of woodpecker. Can we identify the species of tree? Uh, you know, no, I think no, but but I wouldn't be very quick to say that because bioacoustics is a raging field today. And there are all these. I recently somebody sent me an algorithm, uh, uh, that people are developing to just listen to the sound of cough. Someone coughing, and you can actually. It looks like there are ways that you can parse that, and you can say what it may be, like what symptoms it may be to, I mean, leading to some disease eventually. But what I'm saying is that I think that, uh, if you look at it biologically, then certain woodpecker species often have an affinity towards some species of trees. But does the sound of wood sound the same? One could argue that there may be differences, but so far nobody has found some of these differences. But that doesn't mean that that it isn't there or it doesn't exist. So yeah, I also want to kind of since we are at this point, maybe I can connect you to this AI competition that is being conducted, uh, which is in collaboration. That is ISER Tirupati, where I am, in collaboration with Cornell and Google, trying to detect birds with different machine learning algorithms. So do take a look, Pratik, particularly if you're still interested in something like this. So there's a $50,000 award as well if anybody has ML AI skills. Uh, how does genetic data help in conservation? Yeah, that's a good question. Very big picture question about how genetic data helps in conservation. I think again, you can look at this very practically. and you can look at it philosophically. And I'll do the philosophy first. The I think any data helps conservation. Any data that you can add to understand the organisms or the habitat that these organisms live in. It may not seem direct at some point, especially some of the advanced things that often people are not sure how it will help in conservation. But it turns out that they do become important. For example, elephant behavior. The first people who were studying elephant behavior in Africa, they were just tagging individual elephants by just identifying them and studying their acoustics and studying various other things and so on. And a lot of the elephant conservationists did not include them in conservation discussions. But that was a big mistake because we know a lot about elephants. So on that note, I think that any kind of data would definitely be useful for conservation. And I think that segregation should not be there. But genetic data specifically has a lot of value. And most of the value comes from identifying isolated populations, because isolated populations have very specific risks associated with them of extinction and so on. But sometimes you don't know which populations are isolated. Case in point is tigers. I mean, tigers are found everywhere, all across India. But Ranthambore, one of the populations in Ranthambore, has now been identified to be so isolated that it has a lot of kind of inbreeding effects and so on. So much so that the government is considering rescues, like which is getting other individuals in. So this is all genetic data that shows that. And there's other tiger story that's come recently is the Simlipal tiger, which has this black stripe pattern. And again, they say that this is also, you could infer it as something as a sign of some kind of isolated populations. So I think that genetic data, the application and the help for conservation is in how it is applied. You can use the same data and just have only an academic uh, research. But I think many people try to connect it back to conservation, because whenever any data is collected from the wild, uh, often it is, it has value because it can be implemented. So I just need to get to the my slides. Just a moment, please. Yeah, I found some. So here we go. Uh, I think the first thing was this question, which is on uh, genetic data and homoplasy. And I think the question was, if I scroll back up, which have not been explained. Yeah, I can explain. But actually, I was hoping that you don't need them, Akshay. But I can still explain. But I think the primary point is that you have this phylogenetic tree, and you can have a trait which is ancestral, and then it has this one is a derived trait. So all the black are derived traits. And so here in this case, you have two sister taxa having derived traits, and that's apomorphy. And this, I think homoplasy is the simple one, right? Synapomorphy is when there is a derived trait, but because of the ancestor having there's one point where there is a derived trait that gets kind of spread to two taxa subsequently. And autapomorphy is when there is basically apomorphy, but that's just only in one taxa. So yeah, and this here is homoplasy, the example that I was. Because someone asked something else earlier. I'm just trying to find the other thread. Hold on, please. Sorry, I just lost that. Uh, Please hold on. Yeah. But I think I'm not sharing now. Yeah. The other question was about site frequency spectrum. I think you should just kind of, don't worry about too much of the detail. And if you don't get it, but you have these. So if you understood from this what alleles are, yeah, and then what you have to see is how frequently are, sorry, what SNPs are, and what you have to see is, how frequently are these SNPs found in the population, and that is what the site frequency spectrum is about. So you have the rare alleles which are not very frequent, and then some alleles which are common. So if you have a lot of rare alleles, then that means that there is some effect of some gene flow effect or something like that. So site frequency spectrum is one of the basic things from which a lot of these calculations are made. Okay, so there was other question that I actually decided to. Um, Uh, um, I lost the. Yeah, I think it was it was the kakapo. Yeah, that's what made me look for the slides again. Gajanan had asked, uh, in reference to kakapo bird, high incidence of inbreeding led to high homozygosity, leading to inbreeding depression due to deleterious alleles, are in homozygous condition. Is that correct? Uh, I let me just explain this thing to you. Uh, the Kakapo story. Where is that? I think, yeah, that's the Kakapo. So the point about Kakapo is that there are very few individuals remaining, and a lot of organisms. So the whole idea of inbreeding and the problem with inbreeding is that there may be these deleterious alleles, which kind of get fixed through the population. And then, because they are deleterious, you could have some abnormality because of it. That's what is like, essentially what that means. So that can actually be, it can impact individuals particularly negatively, but it may also impact the population. But there is some amazing things that happen in populations, and that's this thing called purging. And that's the one thing that, was there in the slide, this thing called purging. Now, what purging means is that, like these deleterious alleles that are there, let's say that the individuals with those deleterious alleles just die. Um, and it's so deleterious that they just die. But which means that the rest of the population survives. And this is what is called purging. So if you're excessively deleterious, then some of the alleles just disappear from the population. Hopefully, the population there are some individuals which don't have that allele. So that's what it is based on. If all the individuals have, then you're done, right? I mean, that's the classic case. But in some cases, when those are not there, then there is a chance that the others survive, and the ones with the deleterious alleles actually got purged. So just by chance, the ones that did not have it survived, and this process is what is called purging. So you can, inbreeding is bad, but uh, uh, in some cases they don't have deleterious alleles, and that is when scientists like us say, "Hey, no deleterious alleles. Possibly it got purged." I hope that provides some uh, answer. Gajanan, microsatellites are junk DNA, which are non-coding. So do they affect their fitness? No, actually, microsatellites, the thing about microsatellites and a lot of the things that we are trying to look at, we are not looking at the coding areas. I mean, if we were looking at coding areas, that would have some functions which can, any changes would may lead to the things like what I've been talking about. They'll just die. So essentially, the changes that kind of accumulate through genetic drift, those are the ones that we are actually looking at. In fact, when you're studying microsatellites, when you do a project with microsatellites, you take a lot of effort to make sure that the specific microsatellites that you're using are not in Hardy-Weinberg equilibrium and so on. I mean, they have to, as in you remove the ones that are not in equilibrium because that means they're being selected. So you remove the ones that are being selected. So using microsatellites, are good, but because they are neutral, that's the reason why they are good for the kinds of work that we are talking about. Humans are making artificial barriers for species. What could be the consequences of that? Yeah. Again, it seems like a philosophical come reality kind of methods question, but I'll just do the methods this time. I think one of the biggest consequences, there are two actually, maybe three, but big one is habitat loss. So then you have small populations. Second one is fragmentation. So you have isolated population. So you combine the two, loss and fragmentation. That's when the problem comes. So consequence is all of these things that we talked about, low haplotype diversity, low genetic diversity, deleterious alleles, and so on. Mike Deepa says microsatellite short repetitive is it different from repetitive haplotypes? Yeah, in some sense, because haplotypes are like the entire sequence. I can try and give you an analogy. I'm not sure if that will work though. Haplotype is like you say phenotype, right? But it's like that. It's that sum of all things which makes that particular sequence. And if that is identical, then that is what when you say that there is no, that's the same haplotype, no diversity. If it is different, then you say that's a different haplotype. But microsatellite, so the functions of how we use these are also different. Microsatellites are used, what you're trying to do is you're trying to get um, things that change rapidly. And then you're using these microsatellites to kind of look for population level differences across individuals and things like that. So these are very different things. Microsatellites are, you can think of them as markers, but haplotypes are just output of some kind of genetic sequence. Sushila Rathod has said something, but I suppose that's yeah. Sushila, yeah, I already apologized to you, yeah, for that, the song. Naveen says, if partial hybridization happens in strands, is it considered a subspecies? Yeah. So the thing is that the species itself is philosophically a bit difficult to get to because it is something that we created. Because, I mean, and it is an important concept. I'm not trying to say that that's not important, but we want to bin organisms into categories so we know how to deal with them. We know that there is more biodiversity, less biodiversity, and so on. And the main factor there is number of species. So subspecies, you can only imagine that there is more, even more vagueness that is there in how subspecies are created. So I wouldn't say that they did subspecies based on the DNA hybridization thing. But of course, there were subspecies. There continue to be subspecies, and many of them are kind of decided by morphology and plumage differences rather than just the DNA hybridization thing. Uh, in yesterday's session, it was mentioned that it was not known as rufous and white flycatchers, paradise flycatchers were different species, or one was a juvenile form. Uh, Nafisa, can you tell me a little bit about this? I'm a bit, whose session was this?
This was Ashwin's session yesterday.
Okay.
Yes.
Did he
So
say
they,
that, yeah,
huh?
there were different species?
Yeah, he said that there was no genetic data as such that was being conducted to know if they're separate or.
I see. Yeah, I mean, I think that Yeah, that's more a thought thing rather than actual suggestion that they are different species. It's just that uh, there are these morphs that you know that exist, the rufous and white, this one. But yeah, but I don't think that there is really a question of different species. They are well and truly just paradise flycatcher. Uh, how that happens and so on, that's like those are things that we are still beginning to understand. Uh, so how do we differentiate subspecies? I think that subspecies, the idea of subspecies is actually very difficult. Uh, it was created at a time where we didn't have a lot of precise data. So I think that in those times that may have worked well. Uh, because you're saying that, well, these two look clearly different. Let's call them two different species. But then these two, they are kind of different. So shall we just call them subspecies? I think that's what it was like. I'm sorry to caricature this, but the point is that now we are dealing with a lot of data, right? We're talking about thousands of genes, and you can quantify how much difference there is. You have these advanced analytical tools that are there. Uh, Maybe 100 years from now, people will laugh at how we did this or how we are doing this now. But that is just a product of the tools of the times. So it would not be fair to say that the subspecies, you know, are poor in any way. Actually, subspecies do provide a lot of information. In fact, my lab works on the basis of subspecies. So our main currency when we are looking at biogeography is subspecies because we think that a lot of the variation that happens is very nicely captured through these subspecies that are described. I hope that's clear. So yeah, subspecies are important, but the term and how they were created is a little bit fuzzy when viewed with the lens of the data that we have today. And this is not a reflection of what they did 100 years back, but it's just a reflection of how science changes through time. Yeah. Uh, so genetic data has played an important role in tiger conservation in India as well. Oh, yeah, sorry, that's not a question. Yeah, Nafisa, yeah, yeah. Uh, yeah, thanks, Nafisa. That's great. That's great. Uh, which was the reptile at the top of the last slide? Okay, I'll just ignore trivia. Is there a relationship between deleterious alleles and population size? That's actually an interesting question. Uh, One thing that can be said is that the proportion of the deleterious alleles and the impacts that they have will get exacerbated when the population size is small. Yeah. And so the chances of those deleterious alleles having an impact is greater when the population size is small. I hope that's clear. What exactly is next generation sequencing? Hmm, that is one of those things that uh, I think it'll need a lot of reading. Uh, but what I can tell you is this. I just posted a link. So that's you should watch that because that'll give you some idea, I hope. Let me just check that. Yeah, it's how it works. Uh, but I'll just tell you very, very simply. Okay. You remember the ATGC, and you can Sanger sequencing did the very first bit of this ATGC thing where you took just one strand. So essentially, the idea was that you take, like, let's say, blood sample from a bird, and then you extract the DNA, and then you have a PCR, you have a primer. And so primer is a short, like, think of it like a magnet. So you have a little magnet and you have a big DNA. So you attach the magnet, you take the magnet to this, and then so you get this much sequence, right? And that sequence, actually, to get the ATGC, you push it through this thing called the Sanger sequencing, and then you get the entire sequence. But that could only do 600 base pairs or something like that. Now, what Illumina does, I mean, this next generation sequencing does, Illumina is a company that provides one of those thing, and that's the video that is posted there. What it does is that it does like millions of these. They just like found some amazingly clever way of doing this. And so now we are not talking about base pairs, but we are talking about GBs of data that one can generate very quickly from covering large parts of the genome. But does it actually get every single part? No. Then there are different technologies called long read sequencing, which do, so these are only 120 base pairs, but then there are others which do much longer. So there are different technologies within next generation sequencing also. I mean, my goal at least was to just leave you with this idea that things are changing very rapidly, even in how we approach research on birds. And next generation sequencing is one of the ways, one of the broad ways within which there are many different technologies and chemistries that are there. And I don't want to kind of ignore Abhishek Guha's question about Goa gap and Palghat gap. Are these the only two gaps formed in India? Uh, so the question is, what is a gap? And the gap is any break in a mountain. And Goa gap is actually a climatic barrier. So there's no real break in the mountain there. Uh, Palghat Gap is a real mountain break, which is important. But I would think that there are many other gaps. It's just that these two seem to be very significant in Western Ghats. There is Shenkuta Gap, which is further south. And recently, researchers have found that elephant populations have been isolated across a Shenkuta Gap. So I think that there are more gaps than there are mountains for sure. It's just that people haven't studied them. So yeah, that's what I think. But thank you. That's a interesting question. Nafisa, I think we are running out of time, and I have another meeting at 6.
Yeah, sure. Thank you so much for taking the time for this class. I know
Sure,
this
sure.
week's lectures have been
Yeah,
heavy.
yeah. Thanks everybody. I mean, it was great questions, and thank you for engaging. And I hope, yeah, I mean, this particular session and the lecture is a little mm, heavy, but the idea is not that you would create everything. I mean, you would take everything back, but at least some of it, if you can, then that would be useful.
Thank you,
Uh,
Robin. We felt
yeah.
we were taught by a bird, Robin.
Yeah. Thank you. Sure. Yeah. Yeah. The Divya says dodo. Yes, there are efforts to bring the dodo back as well. But yeah. All right. Yeah. Bye. Bye. Thank you.
Bye bye. Thank you so much. And since this is the last set of live sessions for this course, Thank you so much to everyone who's been a part of this course and who's just for taking the time out to attend all the sessions and learn from this course. We've also learned greatly from you all as well. Thank you so much.
Thank you, Nafisa, from all of us. We enjoyed it.
And all the best for the exams. And it's been wonderful interacting with you and getting to hear from you and your questions. Thank you so much, and all the best for your upcoming exams.
Thank you. Hello, yesterday's session is not uploaded.